All right, so functional groups are actually quite in depth. We're talking AP Chem. Um, Mr. Vo informed me that AP Bio also does this, but at least for me, I don't recall ever having to memorize them until organic chemistry, which was sophomore year in college for me. However, we are going to have you guys at least be recognizing and understanding what functional groups are, and then memorize only three, which are listed over here, the hydroxyl, amino, and carboxyl functional groups. So first of all, what a functional group is the specific set of atoms, like CHO, within a molecule that helps determine its chemical and physical properties. These are repeating patterns that scientists eventually picked up after they began analyzing the exact makeup of molecules. As you can see in the illustration, we're circling certain parts of it, and those are functional groups. So while you guys do not have to specifically learn all this stuff, this is definitely O-chem material where you see how carbon and what is drawn down here is essentially carbon. Anytime there's an intersection, you learn after a while that it's carbon. You don't have to know that right now. But in OChem, you look at that and you go, oh, it's going to fold. This OH over here is actually going to connect to the C up there. So there is a functional group there as well as down here. And when they mix it up, they become a new functional group. And we can discuss this in more detail in the future, but you guys actually do have the equipment now. You have the tools to be able to describe what kind of reaction happened, at least on a general level. In a similar vein, this molecule, when folding, actually goes up again to this exact same area here and creates a similar functional group. You'll notice an OCOH. Back here, it was the same thing, OCOH. This in particular is an example of glucose doing the folding. So keep that in mind. Earlier, we discussed how monomers add up to polymers. In this specific case, we have a glycosidic linkage, and here, again, is a specific spot we want you to pay attention to. And now, this should start becoming more and more familiar. You'll notice just now we were talking about carbohydrates. Here instead now, we're talking about lipids. In lipids, you'll see the same thing. Here's another CO and then COH, CO, COH, and here is a bunch of OHs. These are hydroxyl groups, these are carboxyl groups, and what happens to them is you fuse them together and notice that there are some parts missing that used to be there. Again, do a back and forth. You'll notice it was OH, OH, OH in the blue, and now it's just a bunch of O's. You're missing a bunch of H's. On the red side, you have a bunch of OHs that are now missing. And so what happened is you have two H's, one O, and you combine them, they become H2O. This describes to you why we're talking about hydrolysis, as well as condensation reactions, and how you're going to be recognizing this stuff. We can then, from now on, show you this exact diagram. Functional groups really help you understand how these things work together and how to recognize what they are. And over here, probably one of the most important ones, we're going to start focusing on this group on the left. NH2 is the amine group, amide group. I'll have to go look. There's actually a very specific name for it, and I don't want to mess that up. Over here, it's COH and CO double bond O. This is an amino acid. And so when amino acids are combined again to a polymer, you again create H2O. And that bond over there is a very important one that we'll need to be able to recognize in an upcoming skill set. In class, especially for those of you who missed the class, there is a worksheet that's going to be handed out. This becomes a set of notes, one of the few that we're actually handing out on paper. I don't mind if you guys want to do this online. However, the up is that if we talk to Mr. Vo and he agrees to this, we can actually allow this on the test. So I do encourage you guys to practice writing this down. You're going to be not just writing down what the atoms are, but also the functional groups involved and also the atoms and bonds. You should be doing this as a research project with your friends. Go online, see it. You will have to dodge a little bit of bullets sometimes because even though this is fairly common knowledge in chemistry and organic chemistry, I have seen the AI mess up a little time. So when you research, be careful, double check, triple check to make sure it's the correct answer. We'll go over this in class to make sure you have the right ones. Be ready not to write this in pen, write it in pencil so that you can make corrections if need be. All right. Generally speaking, we're giving about, anywhere from 10 to 25 minutes. And so it just depends on what your teacher decides in class. The next part now is to do and learn infrared spectroscopy. Now, we won't actually have the equipment to do the spectroscopy. It's kind of out of our league, unfortunately, but it's not uncommon to find them in, university labs. If we ever make a trip, we might swing by and take a look at one of these. And if we have time, would love to do a sampling so you guys can actually see it. It's a lab technique, and it's used to identify functional groups in a molecule. So, since you guys don't have to do this, we'll just simply explain in general the theory about how it works. So, what you will learn in chemistry in much more detail is that molecules, atoms, they all absorb a certain amount of light and then reflect it off. So, in particular, they discovered that they absorb infrared radiation at specific frequencies, therefore creating a pattern. If you had a liquid filled with water, for example, obviously that would just be water. Just realized what I said, causing them to vibrate and release a wave that the machine picks up. Okay, this means if this radiation comes in, it then gets re-emitted back out, or it gets absorbed completely, or only parts of it gets absorbed, and you get a different color coming out. This is how you can tell from those patterns. Again, this is infrared, so we as humans can't sense them. So we need a machine to do this to help us see, well, what functional groups and what molecules are in there. So in this case, the only real skill you're picking up is that you're going to have to be able to read an IR spectrum data. This is a skill, so paying attention will help you a lot because luckily for you, we're not going to make you memorize any of this stuff. First one up, hydroxyl. In an IR spectrum, you will see essentially a low-level peak. This is essentially just noise because it's whatever liquid's holding it in generally will cause some noise, right? But if you have hydroxyl, specifically that OH over there, and it's connected to the C, you'll get a round peak over here at around 3,200 inverse centimeters. You don't have to worry about anything else. Transmittance, just be able to recognize us. We're not testing you on what transmittance means. We don't even care if you understand what inverse centimeters are, because that's the wavelength, wave number. Right now, just you're going to get a chart in the future that tells you hydroxyl, COH, is at 3,200, and it'll describe it as a rounded peak, right? Then you have ketone, specifically this C, double bonded to an O. Again, you don't have to understand what a double bond is. You're in middle school. We haven't gone over enough chemistry to do this. But again, the noise, and then you have at 1600, around 1600, sometimes it gets a little bit lower, a little bit higher. You won't have an exact measurement, but as long as you're like, okay, it's closer to that 1500 at the bottom there, and it's a sharp peak, now it's a ketone. Carboxyl, also around 3200, but also has this peak. Now, I wonder why these seem so similar. And you'll notice is because earlier it's COH, so that resonates and creates this light for us. Same thing here, and you'll notice there is an OH. Here, there is a double bonded O, so it's at 1600. This can get tricky because you guys will perhaps look at this and go, oh, I remember 1600, it's just this. But if you forget that there's also a 3200 at the end here, you'll have forgotten that this was in fact a carboxyl group. There should be an L at the end there. So it's ketone back here, hydroxyl over there, but if you combine them, it becomes a carboxyl group. You then have the amino group, which is NH2. We mentioned this is a very important group to memorize, and it's this kind of a double peak at 31 to 3400. So 31's over here, 3400's a little bit like that. We'll describe it for you in the chart. But this one, even though we're not asking you to memorize the peaks, you do need to remember the amino group very specifically. Ether functional group number 1. There is a C connected to an O connected to a C in between. Memorize that particular pattern. Well, not memorize, but recognize what it is. And its peak is at 1300. There is, however, a second kind. This is if it's part of a glycosidic linkage, it changes its shape. Now, it has that peak at 1300, but also a 32, because the glycosidic linkage, don't forget, is this with a bunch of those COs. So when you see that, you should be guessing, this is more of a sugar, right? If it's part of an ester functional group, however, and that's the glycosidic linkage you see here, don't forget now there is also a ketone technically, so you get those two peaks. Now there's an alkyl group. An alkyl group is just a bunch of CH, and this one, if you guys recognize from earlier we talked about in biomolecules, is in lipids. So you'll see this almost scratchy peak that comes up, and it's sitting right under 3000 or right around it. So that's where you're going to want to know that this is alkyl. We'll give you a chart that looks something like this. So on the test, you shouldn't freak out and go, oh no, I don't remember any of those peaks. We will provide this for you. However, it is your job to analyze the peaks themselves and be able to recognize what they are. So this is being a skill. The best way to practice this is to go through some practices. Mr. Vo, in fact, has some excellent ones. The way you're going to do it is you're going to read the parasage, look at the actual peaks, and then start to guess. What you should be trying to guess is that over here, for example, it says that the biomolecule is intact polymer, contains a lot of alkyl functional groups. And you notice that because remember back in alkyl, we said it should be around 3000, right? And it's a sharp peak. There you go. It's around 2900. It's a really scratchy, sharp peak. And then the ester linkage contains glycerol and its namesake chains together. Now, because of the other clues you're getting, the 1300, 1600, you check the chart. You're going 1300 CO bond, 1600 is a carbonyl bond, which again, you'll start recognizing. You're like, wait. I think I remember this. I think it's like this, right? So 1300, 1600. Again, you don't have to memorize it. Memorizing would make these easier for you. We're not going to color code them. It's just going to be the same color. But in this case, we wanted to make sure you guys could recognize what we're talking about here, okay? So this is commonly added to bread as a spread before cooking as a coating or as a cooking coating for flavor. Some of you can already look at this and guess, oh, hey, from this guess and this guess, I can probably know what this is. Since you're probably already guessing it's a lipid, as long as you're guessing it's somewhere within the lipid range and it makes sense that you're adding it to bread, sure. It should be that you're guessing it's butter, but I will say that not you knowing any more information, and this is honestly still pretty broad, if you're from a country that's like, oh, we eat like, we put olive oil in our hubs and we add that to like, garlic or whatever else, and that's what we eat. I'll be fine with that as long as it's a lipid. But it's got to make some sense. You can't just be like, oh, I use peanut oil and I spread it all over my ciabatta bread, and that's how we eat it, right? Like I haven't heard that. You'll have to show that to me to get some points. But in general, as long as you're guessing lipids, you should get the majority of the points. The rest of the slides are just more problems with more answers, and Mr. Vo's doing sort of the mystery set, and we can go over these in class. But please make sure to give these a try, because on the test, we will essentially be giving you either one of these or a similar one, just not exactly the ones you've seen here. So with that said, good luck. That's your IR spectrum. Don't forget that you're supposed to memorize the functional groups of carboxyl, hydroxyl, and amino specifically. Good luck.